In this lesson, we'll look at two early experiments on the road to identifying DNA as the hereditary material for life on Earth. We'll begin in the 1920s. In the late 20s, Frederick Griffith was working with mice and some strains of bacteria. He identified a virulent kind of bacteria. That's a deadly strain of bacteria. He called it the S strain because the bacteria had a smooth outer coat. When injected into mice, the mice died. And in fact, autopsy, you could harvest living S strain bacteria cells from the heart. So evidently, the S strain bacteria was deadly. By contrast, there was another related strain of bacteria. The R strain bacteria was non-virulent. It was not deadly. It had a rough coat, so hence the R strain. So S is smooth strain and R for the rough coat strain of bacteria. The R strain injected into the mice did not kill the mice, and there were no bacteria cells found in the heart. Griffith went on then to see if he could destroy the deadly power of the S strain by boiling. When you boil the S strain and inject that material into the mice survived, and no bacteria cells were found in the heart. So evidently boiling can destroy the ability of the S strain to be lethal. But next, Griffith is going to do something interesting. He's going to take some of the dead S strain bacteria, the stuff that was killed by boiling, and mix it with living, non-virulent R strain bacteria. So that mixture turned out to be lethal. The mice died, and furthermore, autopsy revealed living S strain bacteria in the heart. Now, there were no living S strain bacteria injected originally into the mice. There were dead S strain bacteria. The only living bacteria were the R strain. Yet after death, living S strain bacteria cells were found. So let's step back a second. The S strain can kill the mice. The R strain doesn't kill them. Heat-treated dead S strain bacteria do not kill the mice, but dead S strain plus living R strain does kill the mice. Now, did the S strain somehow come back to life in that mixture? No, said Griffith. Rather, what he thought was happening was the R strain bacteria was being transformed into the S strain bacteria. So something from the dead S strain bacteria was getting into the R strain bacteria and transforming them into deadly S strain bacteria. So a chemical from a dead cell can transform a living cell. Transformation then is changing one type of cell into another type of cell. So information must be involved. The transforming chemical is information of some kind, and that's starting to sound like a hereditary type of molecule. Now, in the second experiment, in the 1940s, Oswald Avery is going to pick up from the work of Frederick Griffith. He's going to be using that deadly S strain bacteria, and he's going to ask himself, what could the transforming factor be? What kind of chemical could have the power to transform the R strain into an S strain? Was it DNA? Was it RNA? Was it protein? So he's going to, again, kill the S strain bacteria, heat kill them, and so we're going to break open those bacterial cells, and then you're going to have a fluid, the cytoplasm, with DNA, RNA, and protein in it. And then he's going to take that extract from the dead S strain and put it into 3 tubes. So each of these tubes then has S strain DNA, S strain protein, S strain messenger RNA. And then he's going to add a certain kind of digestive enzyme to one of each of the tubes here. In tube number 1, he's going to add a protein that destroys other proteins. So what will be left in tube number 1 is S strain DNA and messenger RNA. In tube 2, he's going to add a protein that destroys messenger RNA. So what will be left in tube 2 is S strain DNA and S strain protein. In tube 3, he's going to add a protein that destroys DNA. So what's left in tube 3 will be S strain RNA and S strain protein. And now to each of these tubes, 1, 2, and 3, he's going to add living R strain bacteria. So you can see the design of the experiment here. He's systematically destroying one of the 3 possibilities for the transforming factor. This is a nice design, because if destroying proteins eliminates transformation, then we can conclude protein is the transforming factor. If destroying messenger RNA eliminates transformation, then we conclude that messenger RNA is the transforming factor. If destroying DNA eliminates the transformation, then we conclude that DNA is the transforming factor. So what happened? How about that? We get transformation when DNA and RNA are present, transformation when DNA and protein are present, no transformation when DNA is destroyed and only messenger RNA and protein are present. So Avery is going to conclude here that the reason why there's no transformation here is because it's S-strain DNA that is the transforming factor, and he has destroyed it in tube number 3. So for tubes 1 and 2, the transformation took place because S-strain DNA got inside the R-strain, and here it is now, and that S-strain DNA was able to transform these R-strain cells into S-strain cells, the deadly variety. So Avery now has some good evidence that DNA is the transforming factor. And again, since transformation looks a lot like a hereditary sort of information type of change, Avery is going to conclude that DNA is the hereditary material.